This is episode 33 of the Immunology Podcast, Academic Publishing with Dr. Mark Kaplan. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today we have Dr. Mark Kaplan, professor at the Indiana University School of Medicine and editor-chief of ImmunoHorizons, on the podcast to talk about his research and academic publishing. We've also got our usual roundup of recent highlights in immunology news coming up, but first...
Are you looking to stay current with the latest research and news in the fields of immunology and cell biology? Well, we'd like to remind our listeners to check out Stem Cell Science News, featuring the most recent top peer-reviewed research and review papers, as well as industry, policy, and science news. Stem Cell Science News provides a platform that allows researchers to stay up to date with their field while saving time. Subscribe for free at stemcellsciencenews.com. Hello, Jason.
Hey there. How you doing?
Pretty good. Excited about another chat with you about science.
Yeah. Have you gotten to submit or getting ready to submit any papers later?
Not really, but sometimes I know that you have a lot of interesting stories to talk about your publications.
Oh, I do. I mean, I submitted to Nature Communications during the pandemic. That was actually quite interesting. With the you could see the lag as the COVID paper started to hit and being able to get the reviews back.
And everything else was not important.
Well, but then reviewer 3 chilled out. So reviewer 3 stopped asking for more experiments, which was nice.
Oh, nice of him.
Because they assumed you couldn't go into the lab and do any of them, which was true. So it was really interesting to see. You ended up writing like, I cannot do this because I cannot go in. Would you like to accept it as is? And people accepted that, which really makes me wonder how often the game of, I just want to ask you to do something to make you do it is happening. Because you'll see this in papers, right? You'll see like this coherent story with like these 4 extra controls and side thing that some reviewer asked them to test. That's their personal theory about what's going on. That cost the paper a year to come out.
I mean, sometimes you see the delays between first submission and resubmission, and you're thinking, what? That's like 15 months. What happened in there?
I've seen 2 or 3 years before.
Oh my gosh. But, a cynical person would say, well, maybe sometimes they send these papers to people that have similar research interests. And a cynical person would say, well, maybe reviewer number 2 had a similar project going and it's just stalling the progress of the competition. But I don't like to think that. I like to think that that doesn't happen too often.
Yeah, I didn't do the cynical angle. I more did the people like to ask people to do work because if you're a reviewer and you just agree, you maybe haven't done your job. So you always have to find something.
Oh, the review system. Why don't we go into a couple of papers that actually went through the peer review, and they're guaranteed fresh.
Our review of the peer reviewed, guaranteed fresh or your money back. All right. Well, let's see here. I'll do the one that I actually printed out because I read this on an airplane recently.
Old school. I like it.
Old school. Yeah. My poor printer ink, though. Like, man, these color pages. All right. So this one's called Allergen Protease Activated Stress Granule Assembly and Gas German Defragmentation Controlled Interleukin 33 Circulation or Secretion. Excuse me. Its first author's Wen Chen, last author's Bing Sun. It was in Nature Immunology and it came out. It is still in press, it looks like, on natureimmunology.org. So it is just out now.
Okay.
It doesn't have a nice fresh out of press. So I picked this one in part because I studied gas German D during my postdoc. Incidentally, was a gene regulated by what my protein I was studying worked on. And it's the gas German family are really interesting. They call something called pyroposis, which is this pore formed death. And they're generally canonically cleaved by Cas spaces, go to the cell surface and make pores and kill the cell. This is a part of an inflammation in response to fever or IL-1 signaling.
So that's one of the ways the Cas spaces work.
One of the ways, right. But they've learned that the gas Germans are weird and then parts of them signal with other stuff. And there's like there's like cytoplasmic gas German that's signaling, and the other cleave, because it gets cleaved, the other uncleaved part goes and does something magical. And it gets really interesting really quick, and also it's poorly understood. And so this gives another function for gasdermin. So IL-33 is an epithelial-derived cytokine. It depends on response environmental insult. It can drive the allergy response in some cases. What this paper shows is that you have these stress granulin assemblies, and they do this through a stress granule enzyme called, let me pull the name up, that they mostly, PAPin is what they mostly use in the paper. But using mostly cell systems and then some work in mouse lung and then some human samples to kind of translate it. They show that when what's really interesting is that there's gasdermin's usually cleaved to 35 gasdermin D. But I'm just going to say gasdermin at this point because that gets too long. But this gasdermin, because there's A, B, C, D, gasdermin D cleaves to a 35 kilodalton, and that's usually what's involved in pyroposis. But they find that they're in cell death, but they find that there's another cleavage. It's 40 kilodaltons long. And this creates a different pore that is required for secretion of IL-33. So when they're signaling with when they're signaling that says it's time for an IL-33 party, it's made in the nuclei, it's made in the nucleosome area, and then exported to cytoplasm, but doesn't get out. And so they show step one, the stress granule assembly leads to creation, but not secretion. But the gasdermin is needed for secretion. And so they do a lot of cool work. And I think why this kind of has the impact that it does is, one, they have a gasdermin D knockout mouse, which they just got from someone. And I will say boo on the paper in one way, because it just cites and says this gasdermin D was received from this person. And there's no information or citation of the mouse to look up its features.
Uh-huh.
And it's not cited in the paper at all. I was looking for a gasdermin D knockout mouse and never had one, and was actually actively looking to generate one with CRISPR, and it was a mess. So the fact that they have one is interesting. So that is one issue. Now, second, so that's my only ding on the paper. But what they did is they actually mapped out and point mutated. So they made fractional sizes of the gasdermin of different kilodaltons matching the 40 and other ones. Had those expressed, shows that those recapitulated the downstream functions and turning IL-33 secretion differently than the 35 kilodalton size one. And then showed, hey, this is the cleavage region using point mutations, and this is what's really necessary. So they really mapped out, this is the site that gets cleaved. This is the site that causes it. And they also showed it's caspase 1 and caspase 11 independent. So it is not driven by the regular caspase targets.
Is that expected? So under which circumstances do the epithelial cells make IL-33, and which other cells make IL-33?
Well, so a lot of cells can make it. It's usually under epithelial injury, environmental insult.
Okay.
So when it senses macrophages and everything else flooding in because some damage has happened, it'll release it.
But then it would be, so yeah, I guess this means that, because if you have caspase activation, that probably means the cell won't be there much longer.
Right. And so this is a caspase 1 and 11 independent pathway. They don't know what cleaves it, though. So there's something else cleaving gasdermin to this site, but it follows usual putative cleavage binding site patterns, right? So they did analysis, said, okay, these are other cleavage sites could be that aren't the ones we already know about. And they show it's caspase 1 and 11 independent through knockout mice and all these other inhibitors, but you can cleave it, and the cleaved one, if you make it and induce it, does the same thing, and it releases it. And they show it goes to the cell surface and does all the things.
Okay, so this is how epithelial cells
release IL-33 is through a novel mechanism of gasdermin, which has usually been associated with pore-forming cell death.
Very interesting.
Yeah.
What a twist.
Yes.
What a twist.
It opened our eyes to new things, but I'm dumb.
Oh my God. Okay. You know what? I think I'll take over from now with my first presentation. You keep working on your joke, Jay. You're doing great, Jason. I will talk about a slightly different topic, which is immune tolerance. And this really nice short paper from the lab of Mark Jenkins, by the way, a former guest in the show. First author Soon Wook Hong, and was published in Nature on the 6th of July. And its title, Immune Tolerance of Food is Mediated by Layers of CD4 T cell Dysfunction. And basically, it's a very nice overview of looking into the generation of tolerance towards peptides that come in the diet. And he kind of tries to answer the question of why are we not mounting responses against all of these proteins and all of these things that we consume with our diet that are obviously foreign antigens, and we have developed methods to prevent an activation or immune response against the food that we eat. And they kind of look into they have a little battery of 3 different peptides that they give to the mice orally, and they feed the mice, and they look into the T cell responses in these mice, and they characterize as T cells that come up, particularly CD4 T cells that come out of this feeding, and they compare it to the situation in which you have the same peptides that are actually administered together with a cholera toxin that initiates kind of an inflammatory response. And so they start first with a peptide that comes from wheat, that is called from a protein called gliadin that comes from wheat. And they show that basically, if you have mice that consume this protein for the first time, they end up having in the secondary lymphoid organs around the gut and the liver, particularly, you have a small expansion of a subset of CD4 T cells, which they measure with tetramers for MHC2, and they have a kind of there are a couple of different populations that they can identify within this these reactive T cells. They are surprised because they see a small population of cells that are expressing Treg, so FOXP3 positive cells. They see a very small percentage of cells that are expressing follicular helper-like features. Very, very few cells that are expressing ROR-gamma T or TBET or other kind of TH lineage mediators. And they have most of the cells actually develop, they are proliferating, but they're developing into what they call a TH lineage negative cells that don't have any particular, they're not expressing any particular lineage marker. And these cells become kind of dominant in this response. So basically, their protocol, they do 3 feedings. And at the end of these 3 feedings, they see this, and they see this appearance of these cells that seem to not have any particular profile. And so most of them are CD44 low, so they're not kind of like naive, like they don't seem very activated, but they have proliferated, and they can see that their numbers increases. And what is very interesting is that if they keep feeding the mice, so instead of 3 feeds, they do 6 feeds, what they see is basically an increment in the percentage of regulatory T cells, of T cells expressing FOXP3 at the expense of this lineage negative cell. So they see that these cells first become kind of anergic, and then they can transition into a more regulatory T cell phenotype if you keep adding repeated stimulation. So the interesting is that I think what is really interesting is that that they also show that this lineage negative population does not respond very well to afterwards to a different type of immune stimulation. For example, if they do a skin hypersensitivity assay, so the delayed type hypersensitivity assay in the skin with foreign adjuvant, or if they then immunize the mice with poly-IC and the peptide themselves. So they basically end up really characterizing these cells are rather inert and rather incapable of proliferating further, and they become a rather stable population that just don't really that they cannot really very well respond to the peptide long after this initial sets of tolerance, of kind of initial tolerogenic administrations. So, in basically what they show is that although and these populations, particularly the Treg populations, are particularly present in the gut and in the lamina propria and in the colon, and that most of the lineage negative cells are found predominantly in secondary lymphoid organs, and that there's this kind of also separation in the distribution of these cells, which could be maybe related to the fact that cells, if you're having this constant stimulation, maybe Tregs have more access to this stimulation in the gut, and maybe this repeated exposure increases their numbers. And they think that in principle is because of the lack of inflammation that comes together with the feeding with these peptides, results in the differentiation and activation of cells under non-inflammatory conditions with low IL-2 concentrations, so they don't really get to proliferate, to activate very well. They also, therefore, some of them get to differentiate into T follicular helper cell likes, because also IL-2 is not present, which that helps their differentiation. And that basically this generates cells that are rather inert. And this is one of the mechanisms by which tolerance can be generated. What is important to say is that they've been working on polyclonal population, because a lot of the work so far was focused on transgenic mice with single TCR specificities. But in this case, they're working with polyclonal populations that are responding to this peptides of choice.
Interesting. So the paper, though, kind of has a controversial title on CD4 T cell dysfunction. Is this really dysfunction if it's deriving immune tolerance, which can be a good thing? Or is it just what we classically think of as dysfunction is actually something important for tolerance? And maybe we've been branding dysfunction wrong sometimes.
No, because I think this dysfunction is different from like dysfunction in cancer, for example. That is the consequence, usually considered the consequence of like very prolonged antigen stimulation or dysfunction like viral situations. I think this dysfunction comes from a inappropriate priming of the cells to start with, because as the presentation of these antigens are done without with minimal costimulatory signals or IL-6 or other kind of cytokines that induce T cell polarization, and also very little IL-2, these cells, especially I think the low costimulation results in cells that don't get proper TCR costimulation signals, and then they don't develop into fully activated cells to start with, and that we know that can lead to anergy directly, so to say. So I think it's dysfunction in the way that they start functioning as T cells because they cannot really respond to their antigen, but I don't think it's dysfunction in the sense of other contexts.
All right, that makes sense. All right. Well, to keep on the party here, I know we could talk about T cells all day, because
Indeed.
you love them. I'm going to go and talk about the brain a little bit here. I don't even have a good segue, except for thinking about things. I don't know. All right. So this is enhancing GAT3 and thalamic astrocytes promotes resilience to brain injury in rodents. First author is Francis Chu. Last author is Janine T. Paz, Science Translational Medicine. It came out the 6th of July. So this paper goes way more into neuroscience than I usually ever delve, but also has some really cool immune components to it. So there's this thalamic astrogliosis, which is when astrocytes in the thalamus become reactive, and that has changes that reactive changes affects brain function. That's a concept or a thing, and it's a thing that's been seen in brain lesions for people with traumatic brain injury, strokes, seizures, other things. And they're like, well, is this cause or effect? Is this and well, that's not known. We know it's associated with badness, but not that it can cause badness. And so this paper attempts to figure out if it causes badness. basically. So they remind people that it's found in history in postmortem humans with cortical injury, aka traumatic brain injury, and they also show it in a mouse model of traumatic brain injury, which we're just gonna gloss over. They had photoembolic stroke and then controlled cortical impact in a somatosensory cortex, a model of TBI.
We already had that in a paper some episodes ago.
Yeah.
Where they did the
Yeah.
T-Reg salvage.
Yeah, no, Savage is right. So typically a marker is of this occurring is astrocyte marker glial fibrillary acidic protein, GFAP, that it goes up in these cases. And so they did a viral model to try to make a model first. So step one is they show they can make a model that's an adenovirus associated viral construct expressing enhanced GFP from under a truncated promoter of the GFAP. So if GFAP goes up, this will also glow, right? So signs of astrocyte gliosis. But the virus itself causes the gliosis. So they inject it unilaterally into the ventrobasilar somatosensory thalamus. And the cool thing about the brain is if you inject stuff on one side, it doesn't affect the other. So you have an internal control. That's one of the cool things about neuroscience. They show it does indeed cause astrogliocytosis, and that it then they dive into neuroscience like physiology, electromicrocircuit excitability, a bunch of stuff and methods that I only kind of remember from physiology classes, patch clamping, brain slices, and a bunch of things. But if you sum it all up, which is our duties for our listeners here, they show that when you do that, the brain is more excitable from the thalamic astrogliosis, and that can lead to seizures. So if you give mice a drug that can cause seizures, but way lower dose that shouldn't cause seizures, in a control mouse, it doesn't. In these mice, it does. And it can do that for 7 months after you cause the initial injury. So it's a persistent problem for them. Right? So you do the virus, there's no virus left. They have this astrogliocytosis. It lasts a long time and it leads to susceptibility to seizure for a really long time. And so that is the first time that anyone's been able to show that this gliocytosis is causative of seizures. So then they try to figure out how, because there's different levels of tonicity. They go to the GABA pathway, and astrocytes take up GABA through GAT1 and GAT3, and they link all of this through. They see GAT3 expression is significantly down in the injured part of the brains that they cause, and they show that if they induce GAT3, this effect goes away. The susceptibility goes away. Which makes sense given how GABA works in neurosignaling. So they then link that basically astrogliocytosis drops GAT3. That is what causes the problems in the brain. And if you enhance GAT3 in this tissue, you reduce seizures and mortality in a cortical injury model in mice.
But then GAT3 takes up GABA or
right,
releases GABA?
It takes up GABA.
So it would be an excess of GABA because there's no GAT3 because the cells died.
Right. Well, no, they didn't die. They got reactive changes.
Okay. Okay.
And so the reactive changes, there's less uptake of GAT3. So now you're hyperexcitable. And so you have problems.
Oh,
If you take it up and get rid of it, you're less excitable, like your brain should be.
Okay. So that would be a solution to reduce the
post-traumatic brain injury GAT3 shenanigans, or post-traumatic brain injury hyperexcitability. How do you overcome that? Something like this.
And how long do people often, like in human subjects, what is the length of these sequelae when it comes of hyper excitability?
So in mice, it persisted for at least 7 months. We don't know how long it persists in people because we just have brain slices that we look at after they're dead.
But then do you have, because if a person, so let's say a person gets traumatic brain injury and then they get this GAT3 dysregulation, that would cause seizures, or this would cause problems.
So we know that people with traumatic brain injury are more prone to seizures.
Okay.
right? But
Okay.
we didn't know if that's a thing and this injury pattern is a thing, and if they're just both, if anything's causal to anything else. This really establishes the cause that you see with this astrogliocytosis is driving the disease pathology.
Okay. Very interesting. So then to wrap up today, last paper of the day, and now we're moving to B cells. So my A cells are the T cells, but then I get my B cells. That are the B cells. And this paper from Immunity, published on the 12th of July, viral infection engenders bona fide and bystander subsets of lung resident memory B cells through a permissive mechanism. First authored Claude Grégoire, I guess, French, from the lab of Mauro Gascia from the Center of Immunology at Marseille Lumini in France. And I know this, I pronounced it like this because he's Argentinian, so that's how you pronounce his name. So before someone corrects me. So in this case, they tried to characterize and look a bit closer into the population of lung resident memory B cells that come up in response to an airway viral infection, and they have models with mostly influenza virus, and they also sprinkle a little bit of COVID at the end of SARS-CoV-2, just to keep up with the times. And what they find is that, so they try to really differentiate memory B cells, and they define them by B cells that have undergone germinals, that have become part of germinal centers and that have undergone somatic hypermutation. And in order to really identify these cells, they have a mouse in which they have the enzyme, so the gene codes for the enzyme, the aid activation induced cytidine deaminase, which is the enzyme responsible for somatic hypermutation in B cells. That is so the promoter of this enzyme is together with a Cree. They have a Cree system that is also activated by tamoxifen and results in the recombination and expression of YFP. So basically, what they have is a system in which, during a specific time in which tamoxifen is administered to the mice, they can permanently mark B cells that have undergone somatic hypermutation or have activated the hypermutation through expression of AID. And so they have these mice, and basically what they do is they infect them with influenza, and then they add the tamoxifen around between 6 and 10 days after infection, and in which they are looking into marking those cells that are undergoing, that are becoming the memory, that are becoming part of the memory pool of these mice. And so then they look into the YFP cells that come up from these mice, and they characterize these groups. And what they see, and I think it's very interesting, is they see, in the case, they compare B cells that are resident in the lung, so they find in the lung, B cells that they find in the lymph nodes, close to the lung, and also they have some spleen, so those are compared. But I think the most interesting information comes from the lymph node and the lung, and they find basically 3 clusters that they can really nicely model using CCR6 and CXCR3 receptors. And they find basically 2 clusters of cells expressing CCR6, and then one of them also expresses CXCR3. And these 2 CCRC positive clusters are kind of what they really find as kind of memory B cells. Both of these clusters show signs of participation in germinal centers. And they, for example, if you block CD40 ligand interactions, these cells don't form that well. So they really need the whole germinal center shebang. And these 2 clusters, they tend to differentiate towards CD93 positive plasma cells if they take them and they culture in ex vivo in a kind of in vitro situation with feeder cells and activating with CD40 ligand, IL-21, and they end up having kind of these cells migrate towards a plasma cell like a phenotype. So they're mostly they're not making other memory cells or like long-lived memory. They're really right there, ready to make antibody specific, making antibodies upon activation. But then within this CCR6 positive cells, they have what's a separation, which is CXCR3. And here's what I think is very interesting: is the fact that they find that only CXCR3 positive cells really contain what they call bona fide antigen specific B cells. So B cells are actually recognizing viral antigens that they're making, and upon activation, they make antibodies that recognize hematoagglutinin or nucleoprotein from the virus. And this CXCR3 negative subset, they are memory B cells as defined, but they don't seem to be specific against any of the viral antigens. And they have a certain kind of restricted effector capacity, but they're there and they're proliferating, and they become maybe 50% of the overall YFP positive population of like the mutated and chain switched population. So the question is like, where are these cells coming from? Why do you have this huge expansion of B cells that are not being stimulated with antigen? Because if you think of a germinal center, these germinal centers are mediated by T cells that are presenting antigen to the B cells, and the B cells mutate and they react and they receive signaling. But if these cells are not reactive to any of these antigens, where are they coming from? And so they make the point that these are probably B cells that are just close enough to the real memory B cells, to the real antigen-specific B cells, to benefit from particularly IL-4, which is very important for kind of the development of the B cells towards a memory subset. So these B cells become bystanders, and they benefit from the activation environment that the real B cells generate, the real memory B cells. And they suggest that because they express higher levels, they find that they express higher levels of Fc receptors, they might be beneficial and contributing to the overall germinal centers by capturing more immune complexes that have the antigen of the particular antigen that is being presented. And that's maybe how they are contributing, and that's why they're still there, because in the end, they're kind of taking space in the germinal centers. But they might actually be contributing to the activation of the real memory cells by bringing over more immune complexes due to their higher expression of Fc receptors. So in the end, you have, as you also see in the case of T cells, right? You have T cells that are really, you can really detect specificity in their TCRs against the antigens that you're studying. But then there's a bunch of T cells that you don't really know what they're doing there. And there are some of them that you know for a fact that they're not specific against anything in the environment. So it might be that you can see this also in the case of B cells that are resident in the lungs upon airway infections.
Interesting. Were they able to map the distance at which this effect falls off? Like how close does it have to be?
Not really. So they didn't do like very specific experiments. They do say if they inhibit IL-4, if they block IL-4, they have a reduction in these bystander memory cells. So it does seem that it's to be kind of a soluble IL-4 seems to be very important. But they haven't done really like kind of physical separation, look at the physical separation of the cells, because they just see that they're all there, that they also going into the germinal centers. And they show that because blocking germinal center formation through CD40 ligand blockage, that reduces the generation of both of these subsets. So both of them are gaining their activation by somewhat cycling into a germinal center. So they're going to be too far away. Germinal centers are not that big.
It's interesting. More and more crosstalk.
Yeah. Yeah, the complexity is staggering.
It is. All right. Well, we're going to be speaking to Dr. Mark Kaplan in just a moment. But before we get to that, if you're enjoying the interviews on this podcast, we invite you to read more interviews on the STEMCELL Technologies website. In the Immunology Profile series, Immunologists tell their stories, discuss their research, and voice their thoughts and opinions on current topics in immunology. You can find these all at stemcell.com/immunoprofiles.
Continuing our show today, we are joined by Professor Mark Kaplan. Professor Kaplan is chair of the Department of Microbiology and Immunology and the director of Basic Sciences for the Brown Center for Immunotherapy at the University of Indiana. Besides his academic position, he's editor-in-chief of ImmunoHorizons, which is the open access journal from the American Association of Immunologists. Mark, welcome to the Immunology Podcast. Thank you so much for joining us.
Thanks. It's great to be here.
Thank you for being here today.
So I think there's like two basic topics we would like to talk with you today. On the one hand, you have very exciting research on particularly helper T cell subsets. One T cell subset that I found very interesting with these Th9 cells. So maybe we can talk a little bit about this guys, and also about your work as editor-in-chief of ImmunoHorizons that was kind of conceived to respond to a niche or to a kind of a particular area of immunological research. And I think it would be very nice to hear more about that. But maybe we can just quickly start talking about your experience with understanding TH diversity and your role on particularly Th9 cells, which I would say, if I may, that they took a little bit longer to be kind of characterized and their influence in the immune response being better understood. I think like Th1, Th2 were such already for so many years part of the canonical organization of Th responses. And then somehow we missed IL-9 and we missed Th9 cells. So maybe let's just talk about those first.
Sure. So, I guess I can give you a little bit of perspective that I got interested in T helper cells from my postdoctoral work. And when we started working on some of the STAT proteins that are downstream of cytokines that initiate T helper cell differentiation, and worked in that area for a long period of time, and really got interested in IL-9 and Th9 cells about 10, 12 years ago. And we had seen a couple of the papers that came from Gita Stockinger's lab and Vijay Kucher's lab that really kind of reestablished IL-9 secreting T cells as an area. And we were interested because we potentially had a transcription factor that might be contributing to the development of these subcells, and that's what our first publication in that area really was describing Pu.1 as a transcription factor that promoted Th9 cells. I think, as you said, one of the reasons that IL-9 or Th9 cells had been neglected for a long time is that we really didn't know very much about what IL-9 did. And IL-9 was discovered back in the mid-'80s. It was cloned. It had some functions ascribed to it, but in the broader sense of the immune response, it was a little bit more difficult to say exactly what IL-9 was doing. It was maybe promoting allergic inflammation. And those are some of the questions that we've tried to focus on more recently in some of our work.
So to go in a little bit further, I'm a big fan of the gamma delta T cells and the role of T cells in wound healing. And I was wondering if you could dive in a little bit about that. Personally, just because I love understanding immunology, regulation of healing.
Sure. So in terms of healing, I'm really not sure what IL-9 may contribute to the process. There have been a couple of reports that suggest that IL-9 may have some role in immunity in the skin, and potentially in some other areas. But in terms of wound healing, it really hasn't been characterized very well. We did have a publication recently that focused on gamma delta cells in wound healing, and that arose out of our interest in looking at allergic inflammation in the skin and showing a couple of years ago that IL-4, as it promoted allergic inflammation, also seemed to impair the wound healing process in the skin. And one of the things that we discovered was that it actually seemed to eliminate a lot of the epidermal gamma delta T cells from the skin within mouse models anyways. And since that population of cells had previously been shown to contribute to wound healing, that was something that we pursued. And essentially what we showed is that if you block IL-4, then you don't have this loss of gamma delta T cells when you have development of allergic inflammation, and that some of the cytokines that the gamma delta T cells secrete are really important for wound healing. And so in the context of allergic inflammation, if you reintroduce some of those factors, and the one that we worked with in the recent report was FGF7, then you can actually recover that healing response, and even in the absence of gamma delta T cell recovery. And so that's something that could have potential therapeutic applications in allergic patients that have defects in wound healing, that there may be cytokine therapies that could help with that. And that's something that we're looking at a little bit more as to what FGF7 might actually be regulating within the skin, within the keratinocyte response.
Just coming back a little bit, sorry, to the IL-9, because I think we didn't get to that point. So what exactly? So it took us a long time. It was not very clear, but I think now it's pretty clear that IL-9, as you said, has an important role to play in allergic inflammation. But then who are the cells? So we know that Th9 cells make IL-9. We know that other cells also make IL-9. Who are responding to this cytokine? And what makes IL-9 different from other allergy-related cytokines?
Right. And so, I mean, I think that's really a critical question, and that's what we've been focusing on answering. One of the first cell populations that was identified as being IL-9 responsive are mast cells. And I think that they clearly have a role in terms of the IL-9 biological response. But you do have to look at models where you're clearly looking at mast cell dependent functions. And a lot of the models of allergic inflammation don't necessarily require mast cells for their development. What we've described in a couple of papers that have come out recently are that macrophages are one of the other big responding populations, at least within the lung environment. And we showed that both in the context of allergic inflammation and in the context of lung tumor growth. And in both of those situations, the macrophages really seem to be the key IL-9 responsive cell that mediates the immune response. And so in the absence of macrophages that respond to IL-9, you don't get tumors growing in the lung, or you don't get the full allergic inflammation phenotype.
Is there a master switch you found that makes a macrophage IL-9 responsive or not?
No, we haven't. And I think that's a good question. One of the things that we've seen is that it seems to be really chronic exposure to the allergen, and it may be a more chronic stimulation phenotype that leads to IL-9 receptor expression. Within naive mice, we really don't see very much expression. So it's certainly a response to some sort of inflammatory development within the lung tissue. That may be true in the tumor situation as well.
Just for me to understand, in this case, these macrophages, what kind of phenotype do they have? Are they inflammatory or more like tissue repair?
Well, I think from what we've seen, they do seem to have more of a tissue repair. And we don't want to say M2 because macrophage phenotypes are more complicated than that, but they do seem to be shifted more to that spectrum. One of the big targets that we identified as being downstream of IL-9 was arginase 1. And that's one of the classic M2 markers, but we don't see other M2 markers that are IL-9 induced. And it seems that a lot of the biology that we've been studying seems to be as arginase 1 dependent as it is IL-9 dependent. And so it really may be the major downstream factor that we're looking at for function.
So you mentioned arginase, M1 versus M2. How about MDSC, just to complete our circuit here? Do we think there is any MDSC action going on with an IL-9 process? I mean, they're another suppressor, so they shouldn't cause allergies, but God knows.
Right. So in the tumor model, we did look at those. They did not have the same level of IL-9 receptor as macrophages and inflammatory monocytes did. And we didn't see that they were expressing very much arginase either. So, they're clearly active within the tumor situation, but I don't think they're really critical for the IL-9 circuit.
Okay. I think, there's clearly still a lot to understand about the influence of IL-9 on immune responses. And I think it's also very interesting to see it in, and for example, tumor responses, because we always think of IL-9 just like in airways or in other contexts. So I think it's definitely important to look at all angles. But I think that maybe we can do a little switch and talk about your work as editor-in-chief of ImmunoHorizons. So as I mentioned, this is the open access journal from the American Association of Immunologists. And maybe you can talk to us a little bit about, because you were part of the start of the journal. What was the motivation from the AAI to start this journal kind of next to the flagship Journal of Immunology? And what is the need, or what is the space that you're hoping to contribute to?
Right. So ImmunoHorizons started in 2017, and I was part of the initial editorial board, and I became editor-in-chief in 2020. And initially, ImmunoHorizons was conceived to be a capture place for articles that were not, not acceptable or not ready for prime time in the Journal of Immunology. And what we'd seen as a journal before that was that many of the papers that were decided to be not acceptable for Journal of Immunology were then going on to be published in other journals. And we thought, well, maybe we can try and capture some of that segment and keep it within the AAI family, and really have a journal that's geared towards something different. And I think what we've developed is the idea that we're looking for science which is sound, which is performed well, is appropriately controlled, and that the conclusions in the paper are supported by the data that's actually in the paper. But it's okay if it's descriptive, or it's okay if there's no mechanism, or if there's a new reagent that isn't described in great detail, that may be okay too. So I think what we're trying to develop is maybe the model that was originally part of PLOS One, to have any work that is really scientifically sound and it doesn't need to have a major impact. And I think that's what we've been moving towards over the last couple of years. And so one of the things that I think is really important is that when authors submit a paper to the Journal of Immunology, if the editors at the Journal of Immunology decide that it's not appropriate, they will actually consult with the ImmunoHorizons editors, and we will take a look at the paper and decide if we think that it's going to be a good candidate for ImmunoHorizons. And if we decide yes, then the authors, when they get the decision letter from the Journal of Immunology, will actually get an included decision saying the editors from ImmunoHorizons have looked at your article, they think it would be appropriate for transfer, and if you follow the suggestions that they have, that you can transfer your article without too much problem. And I think what's really good about that is that the editors have already looked at it. We really try and say which comments of the JI reviewers need to be addressed and which ones don't. And then if they revise their article and transfer it, then whether it's going to be accepted is really an editorial decision at that point. So we use the reviews from the JI and the responses from the authors, and then the editors are really able to make a decision at that point. And I will say that if an author decides to transfer and they revise the article as suggested, that it is highly likely that it will be accepted to ImmunoHorizons. And I think this is the benefit, that if they were to go to another journal, that they would clearly go into the review process all over again. And so this is not only much easier, but much faster, because we can make the decision based on the reviews that have already been done.
So with this shunt, do you guys also have a shunt earlier on? Like you get the article, it's not going to go out to review because the editor for JI goes, not worth it to even go to review, and then they kick it to you immediately. Is that also a possible path?
It happens, and I will say, the philosophy of the JI has always been we like to review almost everything, because it's a society journal. We don't like to have too many desk rejects. But there are a few cases where something is clearly not going to meet the bar for JI, or it's out of scope of the JI. And yes, they do pass those over to us, and we will take a look at those as well. But if they haven't been reviewed yet, then we will suggest that they could be transferred to ImmunoHorizons, but then they would clearly need to go up for review at ImmunoHorizons, so that there is at least some peer review somewhere in the pipeline.
Just mentioned it a moment ago that, well, of course, we know that both JI and ImmunoHorizons are journals from a scientific society, which is nowadays, given the market and given the journals that are, so many of them belong to companies, for profit organizations. I just have like a kind of a philosophical question to ask. How do you perceive the importance of these kind of journals that are still part of their nonprofit, they're part of their scientific associations? I think you have, AAI, you also have the AAIS, so with science and all the science journals. What do you think the role of these society journals is in today's environment?
Well, I think it really plays a very important role. And I think that money that comes in to support either the Journal of Immunology or ImmunoHorizons, beyond costs of actually producing the journals, goes back to help the society. And a lot of the money that comes into the society goes back to its members. We support a lot of travel awards to come to the AAI annual meeting, but also to other small regional meetings. We provide travel awards to some international meetings. And I think that's, a real benefit of membership, and a real duty of the journals in the field, that we can be part of the society, that we are of the society and for the society. And and I think we're not doing it to just generate money for a company, that it really is benefiting the membership. We have better conferences and better attendance, and and I think the awards are not only monetarily important for a lot of the trainee members, but also it's something that shows that they were competitive for an award, and something to put on their CVs that I think helps them in their careers.
So along those lines, how do you think ImmunoHorizons has been taken to by the market, so to speak, thus far? Like, what's the response been by scientists in the community? Where do you see it going soon?
Yeah, so I mean, I think it's still slowly building, and I think that when we look at the number of papers that are published year over year, it's increasing every year, and so I think that's a positive sign. And I think that we are starting to capture a larger portion of these articles that are turned down by the JI. And I will say that at this point, we're getting of authors that receive an offer to transfer to ImmunoHorizons, I would say about 40 to 50% of them will take that offer. And and I think that's pretty good, but I think that's also a number that we can try and improve over the next couple of years. But I think also trying to get more de novo submissions is going to help as well. And that's something that I think is a little bit more challenging, partially because ImmunoHorizons does not have an impact factor yet. And I think a lot of people really look for that in terms of deciding where they want to submit articles to. And so that's certainly something that we've been working on. And it's a multi-year, multi-process, I guess, to get to the point of actually having an impact factor. But, I think what I can say is that when we do have authors that are publishing in ImmunoHorizons, that they seem to be very happy with the process. It is very seamless in terms of the transfer, and we really try to be very helpful to the authors in telling them exactly what they need to do to fix a paper. And I think that type of instruction is also really helpful to make sure that they're not wasting a lot of time and or money on things that don't need to be done, and really addressing exactly what needs to be fixed to make it appropriate for ImmunoHorizons.
So a streamlined, I think that's very important. A streamlined publishing kind of directions it's very important, because how many people have had a lot of issues getting through all of the different hoops that need to be jumped. For example, as an example of research, your group has recently submitted has a publication, ImmunoHorizons. Maybe you can quickly go over it just to show our listeners what is the kind. I think it was a good example of the type of research that ImmunoHorizons is looking for.
Yeah. So I mean, I think broadly, where as I said, that a lot of the criticisms of research from other journals are exactly the type of descriptors that we want to see in ImmunoHorizons. So if it's descriptive or limited scope, or the results are just not that exciting. So if somebody, for example, generated a knockout mouse and it didn't have a phenotype, then that would be something that could be written up for ImmunoHorizons. If somebody, if a group was looking at a type of therapy and the therapy didn't really have that much of an effect, then that might be something that would be appropriate for ImmunoHorizons. We had an article in ImmunoHorizons a couple of years ago that was comparing a couple of the reporter mice that have been generated for IL-9, and something that was very important to us. And essentially what we did was very descriptive, because we ran the two reporters through a number of assays and looked at which reporter was better in which cell type. And and the results were fairly clear that one of them was actually underreporting for T cells. And that has an impact on how we look at some of the previously published results and what we're thinking of in terms of absolute numbers of what IL-9 positive cells are. So that was important. We had another paper that was published last year where, using the model that we have of atopic dermatitis, we had crossed them to a strain of mouse that was deficient in one of the glycosyl transferases that are important for selectin ligand formation. And what we saw was, not only that there was a protective effect, but that there was a difference between males and females in terms of the protective effect. And that really was very interesting. And that also is the kind of thing that we're looking for in ImmunoHorizons. If you, for example, see a difference in whatever response is being examined between males and females, but you don't know what the mechanism is, or you don't really want to look at what the mechanism is in more depth, then that's something that might be appropriate for ImmunoHorizons. And I think that the challenge is really trying to get people to appreciate what that work that really does have limited scope can be published. We always like to think of, what's the cool story, what's going to be the big breakthrough, and we obviously want to do that, but not everything that we do works out that way. And I think that, for experiments that don't really show what we think they were going to show, or maybe just the results are not that exciting, that can still be published, and I think that still has value to the community. And even negative results can have value because it stops people from wasting time in avenues that might not be fruitful. And I think that we have a responsibility to the community to do that. And I think with NIH now pushing to publish or report all of the data from any studies that you've done that have been grant supported, that ImmunoHorizons might be a destination for that, because we are not looking for a complete story. We're looking for results that, if the experiments are done well, then I think we're interested in publishing those.
Do you have a paper or two or three or however many is appropriate to mention that really stand out in the time that you've been the editor or since ImmunoHorizons came to be, that really like, man, it's great that we had this and look at the impact that it's had since we got it out there?
I think that one of the areas that is maybe underappreciated is the importance of immunology in other species. And, of course, the vast majority of work that's done are mice and humans, but, we have learned a lot of immunology from looking at other species, some from invertebrates. I did my PhD work looking at chickens. And I think that there are a lot of other species that really provide an interesting window to look at how immune systems evolve, very often in parallel, using very different mechanisms for diversification and antigen recognition. And so, some of those articles can be a little bit more difficult to develop mechanism for, and there have been a few that have been published in ImmunoHorizons. And I think there was another study that I was a co-author on, where they were looking at the effects of STAT4 deficiency on development of autoantibody responses. And for the most part, there really wasn't that much of an impact. There was a couple of assays where there was a minor response, but again, not much of a phenotype, but all of the work was really extremely high quality. And so, having a place where that can be published and appreciated is important.
I completely agree that there must be a safe harbor for all of these descriptive work that does not make it to the flashy papers, but I can imagine so many hours of work can be avoided by having this kind of studies in which things such as the quality of a mouse model or the results of a particular genetic mutation or the genetic profile, that they don't give up anything that is incredibly interesting, quote unquote, but that is available for the world. Because, otherwise, as you said, you're bound to do it again. Someone else in some other place in the world will do it again, and they will never know. The answer was somewhere there. I think it's very valuable. So I'm glad that the AI is taking on that responsibility, and I think, as going back to my point about having a society journal, I think as a scientific society that understands the importance of these kind of knowledge, yeah, it makes sense that they do it, and that is really good work.
And as I put out on social media quite a bit, I like to brag that we have the most reasonable editorial board in the business. And I think that we really pride ourselves on making sure that the review process and the decision process is as easy as possible for the authors, and as fair as possible as well.
Well, we like to keep things reasonable and fair, but we also like to ask a fun question at the end here. So I guess the question would be, if you weren't a scientist or a journal editor or anything related to immunology, what would you do and why?
So I mean, I think the alternative career that I've always imagined, and maybe will be the retirement project in the future, is that I always really wanted to be more of a writer, and not a scientific writer, but maybe more fiction, I think. And when I was a kid, I really wrote like police stories and things that were really more creative writing, and that's something that I've always wanted to get back to, and if I had a little bit more time. And as you can imagine, being a chair and editor-in-chief takes up a fair amount of time. But that's actually, one of the fun things is that being editor-in-chief gave me the opportunity to write some editorials that are, I would say, a fair bit less serious than any of the science that goes into the journal. And I've talked a little bit about what goes into the journal, but talked a little bit about some other scientific aspects as well. And and I think that's been fun, and at least getting it out there and having feedback from one or two people that have actually read them, and so that's been rewarding too.
Sounds like a nice future career option. I mean, you wouldn't be the first, right? One of my all-time favorite authors was a scientist turned writer. Isaac Asimov started his career as a biochemist and then ended up being a father of modern science fiction. So I guess that the sky is the limit.
Literally, yes.
All right. So it's been a really interesting conversation. Thank you very much for joining, and we definitely wish you the best on your continuing work at the ImmunoHorizons, and we hope that you keep getting more submissions, and that the journal keeps growing and serving this very important segment, which is a place for all these studies.
Thank you for coming on. That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on Twitter at @ImmunoPodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.